So we've already looked at one reaction where we added HBr to the double bond, and we looked at the spectra for the product and the different peaks, and that spectra is shown here. And so now let's look at another reaction here, right, where we're going to react the same alkene with MBS in the presence of light. And you know that reaction is an allylic bromination. So from that, you're going to get two products because your allylic site here, you're going to form a radical there, but you can also do resonance to get a second radical. But let's look at the spectrum. Right, here's the product, and this is one product. I'm going to have a question in here for you to actually write out and turn in as a bonus question for what the second product would look like. But let's go ahead and look at the spectrum. So here's the spectrum. You can see that there are several signals. We have a signal for this CH3 on the end. We have a signal for these hydrogens. For these hydrogens here, there's another set of hydrogens here. There's one hydrogen here on the carbon that's connected to bromine. There's hydrogens on the CH3. And then there are two hydrogens on the alkene. And both of those hydrogens give off a signal. We're going to talk more about why they give off different signals, so on and so forth. But if you zoom in, you see at 5.37 and 5.06 that those two hydrogens are representative of the alkene hydrogens. And if you remember from previously, we had a chart that said that alkene hydrogens can show up anywhere between 4 and 6, right? But both of those are different, and I'm going to explain why in another example, not in this example, but I'll explain why in a second. All right, and then you have at 4.11 the proton that's adjacent to bromine, and it's a triplet. And then the two alkene hydrogens, I know the resolution is kind of bad, but those are both doublets. So you can see here on the zoom, the red and orange hydrogens that are here, those hydrogens are actually split into doublets. And the reason for that is that they're non-equivalent. I have another slide that I'll show why on, but for now, just know that those two are not equivalent. And so that's why they give two separate signals, and each one of those hydrogens is going to split with the other. And so that's why you see a doublet. So we're going to talk about that more in a minute, right? The reason for that is what we call complex splitting. And so since those two are non-equivalent, they are able to couple with each other, and you follow the same N+1 rule where the red hydrogen is one neighbor and the orange hydrogen is one neighbor. So when you look at a system, the orange hydrogen is going to see the red and is going to couple. So N is 1, N+1 is going to be 2. So it'll be a doublet for the orange hydrogen, and then it'll also be a doublet for the red hydrogen. So we're going to talk more about that later. Right, so you have basically two scenarios where you'll see complex splitting. One is when you have what's called multiple non-equivalents, which is shown down here on the left. And then one where you have hydrogens that are what you call distereotopic, meaning they are adjacent to a chiral center. All right, so here's the rest of the spectrum, where you have the green set of protons here showing up as a singlet for that CH3. And then the purple proton showing up as a triplet, right? Because they have neighbors to the right and then no neighbors on the left, right? So the turquoise protons on the right of the purple ones are going to split with it and cause that signal to be split into a triplet. Same thing here with the other protons that are shown. You can see the teal colored protons are split into a pentet. The light green or lime green are split into a quartet because they have two neighbors on the left, one on the right. And then the royal blue, that's a singlet, and it doesn't have any neighbors. So this is the spectra for the second product. And as a bonus question, if you draw the second product and hand it in to me on Wednesday, which is the next time we'll meet for class, I'll take that and I'll count that as a bonus assignment. All right, let's look at another reaction. It's another allylic bromination. You have trans but-1-enobenzene, and you react that in the presence of NBS and light and carbon tetrachloride, right? So this is the product here on the right. Let's look at the NMR, right? We should see multiple signals. Again, we see some aromatic signals, which are represented by the orange protons and the blue protons. We see the alkene is also giving two signals, and then the hydrogen that's attached to the same carbon as bromine, and then the teal colored hydrogens on the methyl group. So let's zoom in on some of the signals. You see the chemical shifts. There's a doublet at 1.89, and then 4.29 you have a multiplet, and then 6.35 and 6.7, those are your alkene hydrogens. And then the aromatic hydrogens, it's kind of difficult to resolve those. Sometimes they show up as really complex multiplets, but you can see that there are signals, three signals down between 7.24 and 7.40. All right, so let's zoom in on these. Here's my doublet. You can see that doublet is for the methyl group. And then the red hydrogen that's adjacent, that's on the same carbon as bromine, is here. You can see that that's split into a pentet because it has three neighbors to the left and one neighbor to the right. So N is equal to 4 for that. And then the two alkene protons are separate. You see the colors, the green proton and the pink proton. And if you'll notice, they're not just simply split, right? If you notice for the pink proton, it's actually split into two signals. That particular type of split pattern is not normal. It's not your normal n+1 rule. It's actually what we call a doublet of doublets. You can see it split into two doublets, and I'll explain that in a minute. And then the green proton is split into a doublet. So the doublet of doublets is because the pink proton is coupling or splitting with the red proton to its left and then the green proton to its right, right? So those two, N is 1 in each case. And so you're going to have N+1, which is 1 plus 1 equals 2 for the red proton, for the splitting with the red proton. Then you'll have 1 plus 1 equals 2 for the splitting with the green proton on the right. And so because of that, that signal is split into what we call a doublet of doublets. And then the green proton is on the alkene also, but it's only split into a doublet. And then if you look at the aromatic section, it's really messy, but there are actually like three signals down there. One signal for the two orange protons, and then one signal for the two blue protons, and then one signal for the one lone black proton that's at the 4th position on the ring, shown here.